Hello, junior microbiologists. Just checking to make sure my pen works. So this is the second video in a brief history of microbiology. So it's chapter 1 from Bauman. So we, in the overview, we looked at this flow chart of the biologists and the modern disciplines. So we have the early years, so pre-1857, and then the golden age of microbiology, 1857 to 1907. And then all these scientists that worked within the golden age. So there was the early age, like we said, we talked about the the early age, pre-1857. That's Leeuwenhoek, Linnaeus, Semmelweis, and Snow. And then the golden age are all these guys, including Pasteur, which everyone's heard of. And then the modern age is 1907 to the present. And then there's the future age. So that's now into the future. So the ages of microbiology. But basically, 4 individuals changed the way healthcare was delivered in the 19th century. So since most of you are going into healthcare, probably be interested in those 4 individuals. That would be Ignaz Semmelweis, my hero, Snow, Lister, and your gal, if you're going to be a nurse, Florence Nightingale. So the pre-golden years, this is a drawing of Robert Hooke, who was the first guy to coin the term cells. So here's a picture, here's 2 cells. Here's one cell with its nucleus, its cheek cell. Here's another cell with its nucleus. So 2 cheek cells. Robert Hooke used this primitive microscope in the 1600s to view cells. So he was the first guy to use that. Now this guy is interesting. Antony van Leeuwenhoek. These are the years that he lived. He made these really sweet microscopes, and he would have parties and show people all the things you could see with these cool little microscopes. But then he never shared his information on how to make them with anyone. So thanks a lot, buddy. So this is kind of a recreation of what he made. So here's the lens. Here's the specimen holder. And then I'm not really sure how they work because I've never really seen one, but apparently you could see some microbes with these at his parties. So he's the first person in history to view microbes, including bacteria, fungi, and protozoa. He called the microbes animalcules, which is kind of interesting, kind of like a little animal. He made excellent microscopes, but like I said, he kept the secrets of making microscopes to himself and never taught anyone. So thanks a lot, Antony. So we talked about the 4 questions in the overview. The previous video, the scientists were asking 4 questions, important questions, in the golden age. And the first question was, is spontaneous generation of microbial life possible? So spontaneous generation just means living things could just come out of nowhere from non-living matter. So something living could come off of a brick, for example. So philosophers and scientists in the olden days thought living things came about in 3 ways. So spontaneous generation, I think Needham was the first person to use the term spontaneous generation, but Aristotle had the first notion, at least written down notion, of spontaneous generation. He called it abiogenesis, but it's the same thing. Spontaneous generation, abiogenesis is the same thing. Because Aristotle thought maggots arose from meat. So those are just, maggots are just, I think they're the precursor to an adult fly. Living things could also arise using sexual reproduction, which is what we use. And then living things could also come about by asexual reproduction. So this guy named Red Eye R-E-D-I, or ready, he did an experiment to test Aristotle's hypothesis that maggots arose from meat. So he had these 3 flasks. He might have done more, but in this cartoon, this is what it's showing. So here's a flask, and here's a piece of meat, delicious steak, and he left this flask unsealed, and he found that maggots and flies came from the meat. So if he just did this experiment, he'd say, oh yeah, Aristotle is right. Here is another flask with meat inside, and it's got a cork top. The flask was sealed, and no maggots came about. Then he did this really cool experiment where he put the meat in the flask as usual. Then he put this piece of gauze around the top, and I guess the flies could smell the meat somehow, and they found maggots on the top of the gauze. So Francesco Redeye provided evidence against spontaneous generation through experiments with meat and maggots and flasks. So Pasteur also did some work with this thought, Pasteur's experiments and the scientific method. So you probably learned the scientific method in 201, I'm guessing, or maybe 156, or Bio 100, any of those. So scientific method, first thing you do is you observe things. That's what these scientists were doing, and then they formulated a question. From the question, they turned that question into a hypothesis. I like to form my hypotheses in a logical if-then format. If this happens, then that should happen, and then you can test that with an experiment. So then after you have a hypothesis, you can do an experiment, including control groups. Those are the groups that don't get the treatment of interest. And then after you do your experiments, you make observations, and your observations will either support your hypothesis or they don't support your hypothesis. If the data or observations, the data from the observations support the hypothesis, then you accept the hypothesis, and if a whole bunch of people do a bunch of tests about the observation of interest, and their hypotheses are all correct, then you get a theory or a law, like the law of gravity or the cell theory that every living thing is made up of cells. If your data don't support your hypothesis, then you reject it. You are rejected hypothesis. And then you can also modify your hypothesis here and go back and repeat. So rinse and repeat. Go back through the scientific method. So Pasteur did these experiments to disprove spontaneous generation. So he had this is a flask. So this is the bubble part of the flask. And then this part here, this is called a swan neck flask. So if you've ever seen a swan, maybe you know why they call it a swan neck flask. So they've got this infusion, this stuff in here, this brown stuff. I'm not sure what it was. Maybe some kind of like when you boil meat and then you take that water that was boiled in the meat. That's the infusion. But it's there, so if there's anything that can live, needs food, then it will be able to do that. So they heat the infusion. So boiling, in their minds, would kill any living things that got into the infusion somehow. So now you've got just this pure infusion with should be no microbes in it. And then the steam from the boiling will escape through the swan neck flask. And then they let these flasks sit for a long, long time, and you didn't see any microbes. So where's the spontaneous generation? There's stuff to eat. There's air able to get in and out. Where's the microbes? I guess there's no spontaneous generation. But then they went on to do some more tests. So here, the air with the dust in it and the microbes in it will get into this part of the swan neck flask, but it'll settle here and it won't be able to move up through the swan neck flask. That's the swan neck part of the flask into the infusion. So, and I'll show you what he did in a sec, but Louis Pasteur is considered the father of microbiology. He proved through these experiments with swan-necked flasks that microbes arise from other living microbes, not from non-living material. And we talked about this. So the swan-necked flasks were left open to the air for up to 18 months, and nothing grew there. So no spontaneous generation. But then, this is kind of cool, this next slide. After 18 months, he broke off the necks and let that dust that was in the part of the swan neck fall in, or he tilted others so that the liquid touched the dust, and then the microbes grew. So that's pretty cool. The microbes are in the dust. They were not spontaneously generated. So after 18 months, here's the cartoon that goes along with that. So the flask, now the infusion that can feed microbes, gets up here to where the microbes are. And then the microbes now have something to eat, so they can start growing, reproducing, and microbes appear in the infusion. Spontaneous generation disproved. Second question they were interested in at the time, because a lot of, well, rich people, I think, that were making wine hired Pasteur to figure out what causes fermentation so they could make more money by making good wine most of the time instead of bad wine. Sometimes the wine spoiled. Why did it spoil? First, Pasteur had to find out what caused fermentation, and he did. He found out that the yeast, Saccharomyces cerevisiae, will ferment the grape juice to make alcohol. And we'll look at the picture of that in a sec. So he developed a method that would kill the spoilage organisms, and he wasn't sure what they were, but they were bacteria, but not kill the flavor of the wine by boiling it. This was the first instance of pasteurization. The field of industrial microbiology was born. From this work, Pasteur hypothesized that disease in people was caused by microbes. So if diseased wine, so make bad wine, can be caused by microbes in people with a disease, the cause could be a microbe. So let's take a look at what causes fermentation, Pasteur's experiment. So here he's got his flask. Here's the fermenting grape juice. And if you do a microscopic analysis, this shows juice contains yeast and bacteria. So the yeast is actually what ferments the grape juice into wine, and then the bacteria can spoil the wine. So let's look. His first hypothesis is spontaneous fermentation occurs. So the experiment that he did, so this is day one. The flasks of grape juice are heated sufficiently to kill all microbes. So here's a little flame here. The flask is heated, and then it's sealed, and then on day two they seal it. The observation they make, there's no fermentation. So that disproves spontaneous fermentation. So remember, spontaneous just means it arises out of nowhere. The grape juice was not able to be fermented into wine because they killed everything in it, including the yeast, and then they put a cork on it so nothing could get in. So you reject the first hypothesis. Second hypothesis, air will ferment the grape juice. So there he gets it out of swan-necked flasks, so the air can go in and out. They boil it again. Here's the little fire. The flask remains open to air via a curved neck. Didn't get any fermentation, and there's no microbes in the juice. So you reject the second hypothesis. Third hypothesis, the bacterial microbes ferment grape juice into alcohol. So again, here's their flask. They heat it, but then they inoculate the juice with bacteria, and they seal it so nothing can get in. Bacteria reproduce in the juice because there's grapes in there, and they can use that to live and reproduce. And they make acids, not wine. So they modify their hypothesis. The bacteria will ferment grape juice, but not into wine. Bacteria ferment grape juice into acids. Then the fourth experiment they did, so they heat up the wine in the flask, and then they inoculate the juice with yeast, and they seal it so no bacteria or anything else can get in. The yeast can grow and reproduce, and you make alcohol. Woo! We'll accept the fourth hypothesis. Yeasts ferment grape juice into alcohol. And you remember when he put the bacteria in the grape juice and then put a cork on it? When he put a cork on it, that means no oxygen could get in, but the bacteria were still living. That makes them anaerobic bacteria. And we'll talk a lot about aerobic and anaerobic bacteria in the future, but because these bacteria could live in the flask with no oxygen coming in, they're anaerobic bacteria. And he showed they cause the wine to spoil by making acids. So Pasteur's work. So now, oh, I'm sorry, let me back up. Now we're at question 3 that all these people were asking in the before times. What causes disease? So Pasteur's work that he did, along with Koch's postulates that we'll look at on the next slide, gave birth to this germ theory of disease. So before the germ theory of disease, people just thought you were possessed by a demonic spirit when you got sick. Some people thought that. They had all sorts of theories about why you got sick or why you had a disease. Pasteur and Koch together, their work gave rise to the germ theory of disease, stating that microbes cause infectious disease. So Pasteur's work, along with this guy Koch and Koch's postulates, showed that microorganisms cause disease. So let's take a look at the cartoon. Let's start up here at number 1. It says the suspected germ or microbe must be present in every case of the disease. So check it out. Here's our diseased subjects. So they are rats. And they all have the same disease. And check it out. Every one of these rats, they isolated the same bacteria, and they got that bacteria and they smeared it on a plate, which you guys would do in the lab. Hopefully we'll do it virtually. And they could grow that bacteria into a colony. So a colony is a whole bunch of bacteria living together. And for example, this red lump. So this bacteria grew this red colony. Same thing here. This bacteria grew this red colony, and this red. Any of these red colonies are colonies of this bacteria. And same with the third diseased rat. So Koch's postulates. Number 1, first postulate: the suspected germ or microbe must be present in every case of the disease. The second postulate: the germ or microbe must be isolated and grown in pure culture. So this is the first plate that they did here. They grew a bunch of different colonies, but in every one of these plates from every one of these rats, they found this red colony. And then they took a little bit of that red colony and they smeared it out here on this other plate, and it grew. So this is a streak plate, which hopefully we'll be doing virtually. But this is a pure culture, and it's a pure culture because it's only got one microbe in it. So these plates here, they've got more than one type of colony, so there's more than one type of microbe. These have only one type of microbe, so they're pure culture, and they're called streak plates because you take the inoculating loop and streak it back, and well, actually you dip the inoculating loop in this colony and then you streak it back and forth to make a streak plate. The third of Koch's postulates here, it says the cultured germ or microbe must cause the disease when it's inoculated into a healthy host. So then they could take this microbe, put it in a syringe, inject this rat would get sick, and then they could re-isolate that same microbe and grow it on a pure culture. That would show you that the original microbe caused the disease. And then also they had to show over here that that germ or the microbe was not typically found in healthy subjects. So in this rat, you didn't find the disease-causing microbe. So in summary, Koch's postulates are criteria that people use to establish that a specific microbe can be shown to be the cause of a specific disease. These postulates can't be used to prove the causes of all disease, but if it's a single microbial disease that you could use to prove it. And then here's the Koch's postulates written out. Koch discovered steps to prove the causative agent of disease, and these are the 4 steps, and we went over those. But you can pause the video and look at these and write them down if you want to. Koch also studied anthrax. You probably, I don't know if you're old enough to remember when there was the anthrax scare in the early 2000s, but he studied anthrax and was also involved in developing the germ theory of disease, along with Pasteur that we talked about. These were his experiments with anthrax, and then he also did some experiments with diseased humans. So you can read through those. This is kind of cool. This is a modern agar plate, and it just, I put this up here because if you, let's say, took some, like, you know, the swabs you use for your ear, and you put that on a toilet seat, and then you swabbed it onto this agar plate, you'd find all these different types of bacteria growing in these colonies. So for example, this is bacterium number 4. This is not a single bacteria. This is where people get kind of confused. This is a bacterial colony. There's probably millions of bacteria number 4 in this colony. It's a modern agar plate, but Koch used a potato to grow bacteria on. Genius. This is a gram stain. This is like basic microbiology 101. You guys, if you were in the lab, you will learn this. But if you're doing this online, hopefully we'll get a virtual gram stain. But these are microbes, and if you are a gram-positive microbe, you'll be really dark purple like this. So each one of these is a little coccus or a ball. That's gram-positive, will be purple. If you are a gram-negative microbe, you will stain pink. So it'll look like this. And those look like rods. The guy who developed this, Hans Christian Gram. If you just put a microbe on a slide and looked at a microscope, you wouldn't see it. Microbes are colorless. So Gram used a series of dyes that stained some bacteria purple, that's gram-positive, some bacteria pink, that's gram-negative. It's the most widely used staining technique when you're looking at bacteria. He used this because, if you remember, to fulfill Koch's postulates, you have to see the microbe. And Gram was like, well, this is how you can see a microbe. Stain them. Here's a light micrograph of a Gram stain. So you can see that this is a mixed, this would be a mixed stain because you've got these pink rods, which are gram-negative. So here's a pink rod, here's a pink rod. And then these purple balls, a coccus, those are gram-positive microbes. Okay, now we get to my hero, Ignaz Semmelweis. So this is a photograph of, in the old-timey days, I don't know, 1500s, I'm not sure, a woman giving birth. So she's sitting in this chair, she's got a birthing chair, and this midwife, I guess, has got her hands up between the woman that's giving birth, between her legs to help birth the baby. That looks really, really terrible. But anyway, it's an old-timey photo. Women would give birth, and sometimes they would get this puerperal fever when they gave birth, and then they would die. And people were like, oh, why are these women dying. They're getting this fever after they give birth, and then they die. This is where Semmelweis comes in. So we talked about this earlier. 4 individuals changed the way healthcare is delivered. Semmelweis, Lister, Nightingale, and Snow. They did all their work in the 19th century, so the 1800s. In the mid-1800s, they didn't really do any sewage treatment, didn't have good hygiene, nosocomial infections were rampant. Nosocomial just means a hospital-acquired infection, and they're called hospital-acquired infections, or HAIs, hospital-acquired infections, but this is from an older book, so they call them nosocomial infections. Surgical patients, you go into surgery, they would die from gangrene. Women giving birth died from this puerperal fever, like I said. There he is, my hero, Ignaz Semmelweis. He lived between 1818 and 1865. He was an obstetric ward doctor in Vienna. So what he did, he observed his wing where medical students, they would train on cadavers. So they go into the cadaver room, they cut the cadavers up, look at the tissues of the cadaver, and then without washing their hands, they would go into the birthing wing where midwives were assisting mothers with birth. So you go from the cadaver room to a place where women are giving birth, and when women are giving birth, there's a lot of blood and there's a lot of torn tissue, and these doctors are touching these women with torn tissue. Ignaz saw that where that happened, 20 times more women were dying in childbirth than in another wing where the young doctors in training were not going from the cadaver room to the birthing room. And he hypothesized that there were these things called cadaver particles. They were microbes, but he called them cadaver particles that would result in puerperal fever in these women. They were carried from the delivery room to the delivery room from the autopsy room. I said it was a bacteria. Semmelweis was awesome. So he did a cool experiment. He required his medical students to wash their hands in chlorinated lime water to prevent puerperal fever. Mortality went down from 18.3% to 1.3%. Amazing. But of course, he was ridiculed by the other doctors there. That, oh, they don't need to wash their hands. Anyway, he was forced to leave the profession, and he died in an asylum, some say, of the same bacteria, the same fever that the women he saved had. Tragic tale. Joseph Lister, 1827 to 1912. He noted that there were a lot of infections after surgery. So here's some old-timey doctors doing some surgery. And he saw that after surgery, some people got a lot of infections. Yeah. So he developed antiseptic techniques. So his great discovery was antisepsis. He had his assistants spray wounds on the patients, the incisions and dressing, with this carbolic acid or phenol. And this is the unit that he used to spray with this to kill the microbes. Of course, the medical community was like, oh, you're an idiot. But, so his technique was met with resistance, but death went down by 2/3. Amazing. Here she is, Florence Nightingale. Here she is in the military hospital. There's an actual photograph of what she really looked like. 1820 to 1910. She introduced cleanliness and antiseptic practices into nursing. That's pretty cool. She also pioneered military hospital reform. So she said, let's keep this place clean, people. And she also pioneered nursing education. Let's educate these nurses before they come to these battlefields. She scrubbed the hospital. She changed the patient's dressing. She documented statistical comparisons. That's pretty baller back in the day. Got to give her props. She founded the first nursing school. She is your hero if you're going to be a nurse. Okay, here's our guy, John Snow. Remember, there's 4 people that changed the way the way hospital settings ran. John Snow, 1813 to 1858. The Broad Street Pump, he studied propagation of cholera in London. You probably heard of cholera. Anyway, he basically is a father of epidemiology because he made a map of where everybody was getting sick, and he located this Broad Street Pump, and he had a really sweet map of all the illness rates and the death rates around the Broad Street Pump. Here's a photograph of the actual Broad Street Pump that people were going to and getting cholera from. Okay. He had the city shut down the pump. He must have been powerful. They shut it down. Cholera levels went down. And he started epidemiology. So we're still on question 4 now. Let's look at, now we're on a new question. How can we prevent infection and disease? So Jenner. This is a painting of Edward Jenner giving the first inoculation of cowpox into this boy. There's different stories. I originally heard the story that he made one of the women who worked for him, made her give up her son to be tested on. I don't know if that's true or not, but anyway, he tested the hypothesis that cowpox provided protection against smallpox. So smallpox is a terrible disease, you can die from it. But these milkmaids that worked with cows would get this milder form of smallpox called cowpox. So he would take the scabs off of these milkmaids who had cowpox and grind them up, and then he injected those cowpox scabs into this boy here in this painting, and then he was exposed to smallpox, and he didn't get a really serious case of smallpox. So this Edward Jenner invented vaccination, basically, and the field of immunology. So he developed the first vaccine against smallpox. And vacca is Latin for cow, so that's where we get the word vaccine. So the milkmaids had cowpox, they didn't get smallpox. Okay. Paul Ehrlich, so we're down here in our chart, he invented, well, his work led to the modern discipline of chemotherapy. He wanted to find a way to kill microbes but not kill our cells. So kill the microbial cells, but not kill our human cells. He wanted to find a magic bullet that would just go in and target the microbes and not target our cells. He's father of chemotherapy. That's a medical branch of microbiology. He never found the magic bullet, but he did find a chemical that was effective against this protozoan that causes sleeping sickness called trypanosome, and against my favorite microbe, just because of the name, Treponema pallidum. That's a causative bacterial agent of syphilis. And he found this chemical, he called it salvarsan, and he used it to treat syphilis. Other notable scientists in the golden age of microbiology, so you can check this out, but I think that's the last